Thanks for tuning in to Planetterrian Daily. It's March 26th, 2026. Here's your science and health briefing. Let's begin with a genuinely elegant new approach to killing cancer cells that feels different from anything we've seen before. Researchers in Germany have created a light-activated copper-based agent that triggers a form of cell death called cuproptosis. This mechanism itself was only formally described in 2022, which tells you how fresh our understanding of it still is. When the compound is hit with light, it forces malignant cells to take up and accumulate toxic levels of copper inside them. In laboratory tests, the technique proved dramatically more effective at eliminating cancer cells than standard chemotherapy agents. What stands out to me is the precision. Traditional chemotherapy attacks any rapidly dividing cell, which is why it creates so many harsh side effects. This method, by contrast, homes in on a specific metabolic vulnerability that cancer cells appear to have around copper regulation. Copper is essential for life. Your cells use it every day for energy production and antioxidant defense. But too much of it, delivered at the right moment and in the right place, flips a switch to a distinct programmed death pathway. Early data suggest a single cancer cell can be killed with roughly 1/100th the dose compared with conventional agents. That's the kind of therapeutic window researchers dream about. It makes me hopeful we might one day treat certain hard-to-reach tumors without punishing the rest of the body quite so severely. Of course, we're still in the lab stage, but the biological logic here is clean enough that I'll be watching closely for the first clinical translation steps. While this new cancer approach targets an intracellular metabolic choke point, another team has zeroed in on a different kind of cellular housekeeping failure that appears central to Parkinson's disease. Scientists have identified a protein called TMEM175 as the key ion channel that keeps the right level of acidity inside lysosomes. Lysosomes are essentially the cell's recycling centers. They break down damaged proteins and other waste so it doesn't accumulate and poison the cell. When TMEM175 doesn't work properly, the pH inside these compartments drifts, the recycling process stalls, and toxic material starts to build up. That dysfunction is now strongly linked to the cellular problems seen in Parkinson's. The discovery is exciting because it gives us a concrete molecular target rather than just treating motor symptoms after the damage is done. For years, we've known lysosomal dysfunction was involved in neurodegenerative diseases, but pinning down the exact regulator of acidity has been elusive. This work suggests that maintaining the correct pH balance inside these tiny compartments may be far more important than we previously appreciated. It's the kind of finding that doesn't immediately hand us a cure, but it sharpens the focus for drug developers and makes future therapies feel more biologically grounded. I find it quietly encouraging. Sometimes the most powerful advances come from understanding the housekeeping machinery we've been overlooking. Staying with the theme of cellular function and decline, there's a fascinating new study that uses fish as a model to ask when aging actually begins and how we might spot it earlier. Researchers tracked fish over their entire adult lives and discovered that sustained activity levels and consistent nighttime sleep patterns during midlife were strongly associated with longer overall lifespan. Interestingly, aging didn't look like a slow, steady slide. Instead, it progressed in relatively sudden stage-like jumps. One period of relative stability would be followed by a fairly abrupt shift into a new phase of declining function. This pattern makes the work feel relevant beyond the aquarium. It suggests that the biological switches between aging stages might be more discrete than we thought, and that everyday behaviors in middle age could serve as early warning signals. The idea that we might one day track simple daily patterns in people to anticipate later health problems is compelling. Of course, fish aren't humans, and we have to be careful not to over-extrapolate, but the study adds to a growing body of work showing that midlife is a critical window for long-term health. It leaves me wondering what equivalent markers we should be paying closer attention to in our own lives well before obvious frailty appears. From these insights into how cells and organisms age, we shift to the lingering effects of viral infection. Single-cell analysis of blood samples has revealed a distinct molecular signature in white blood cells of people experiencing persistent fatigue after COVID-19. The signature appears particularly common in those who had initially mild or moderate infections rather than the most severe cases. This provides one of the more concrete immune clues we've seen for a condition that still affects a significant portion of people post-infection, with estimates suggesting it touches roughly 1 in 10. What's striking is how specific the cellular changes look under high-resolution analysis. It's no longer just a vague something's off with the immune system. Researchers now have a clearer molecular target to understand why fatigue can linger for months or years. This kind of mechanistic detail is exactly what's needed before we can design rational treatments rather than just managing symptoms. Long COVID has been a humbling reminder of how much we still have to learn about the long-term consequences of even seemingly straightforward viral infections. Any study that moves us from description toward mechanism feels like genuine progress. Let's move from the very small to the very large. Cosmologists in China have put forward a mathematical model that allows dark energy to evolve over time while also addressing the so-called Hubble tension. For years, different methods of measuring the universe's expansion rate have given conflicting numbers, and that discrepancy has been one of the most stubborn problems in modern cosmology. This new framework offers a way to reconcile some of those conflicting observations without throwing out the standard model entirely. It's not a complete revolution, but it represents a thoughtful attempt to let the data guide refinements rather than forcing everything to fit a single fixed assumption about dark energy. Closer to home, astronomers have solved a more local puzzle within our own galaxy. Detailed computer simulations now show that the Large Magellanic Cloud, one of the Milky Way's satellite galaxies, is acting like a piston. As it moves through the surrounding gas, it compresses hot material in the southern part of the galactic halo, making that region noticeably warmer than the northern side. The finding neatly explains a temperature gradient that had puzzled researchers for some time and improves our understanding of how smaller galaxies can influence the larger systems they orbit. It's a nice reminder that even in a galaxy as vast as ours, relatively small neighbors can leave measurable fingerprints. Back on Earth, researchers have been studying water movement through the so-called ghost forests along the eastern U.S. coast. These are former coastal woodlands that have been killed by saltwater intrusion as sea levels rise. By examining how water and nutrients cycle through these dying ecosystems, scientists are gaining insights into how coastal forests respond to climate-driven changes. The work isn't dramatic, but it's practical. Ghost forests serve as natural laboratories for understanding the future of coastal resilience. The more we understand the transition zones between forest and marsh, the better we can plan for vulnerable areas in the decades ahead. In a very different kind of environmental research, a team at the University of Michigan discovered that common laboratory gloves shed microscopic particles that can contaminate samples. Both nitrile and latex gloves release tiny bits of material that get counted as microplastics in environmental testing. The finding is a useful reality check. As we try to understand the true scale of plastic pollution, we need to be rigorous about our methods. This study highlights how easy it is for lab contamination to skew results and why careful controls matter. It's the kind of quiet methodological work that doesn't make headlines but improves the reliability of an entire field. Before we wrap up, I'll just say that the cupraptosis story is the one I'll be following most closely in the coming months. Watching how researchers move these light-activated copper agents from petri dishes toward early human trials should be fascinating. The idea of using a metal our bodies normally need and carefully turning it against cancer still strikes me as quietly brilliant. That's Planetterrian Daily for today. If you enjoyed the episode, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. This podcast is curated by Patrick, but generated using AI voice synthesis of my voice using ElevenLabs. The primary reason to do this is I unfortunately don't have the time to be consistent with generating all the content and wanted to focus on creating consistent and regular episodes for all the themes that I enjoy, and I hope others do as well.